you see this okay?
Yes, we can see it very well. Thank you.
Okay, perfect. Thanks so much, David, for the introduction and for inviting me today. So yeah, just to give you a little bit about my background, I'm actually originally a chemical engineer by training for my undergrad degree, and I did my PhD at the University of Washington just down the road in bioengineering. I pivoted for my postdocs. I actually did my postdoc in a pure immunology lab where I was working on self-amplifying RNA vaccines, including a first-in-human siRNA vaccine against COVID-19. So I'll talk a little bit about this today, but just to give you kind of an overview of where I come from. So I think I fit well within this biomedical engineering department because I really come from both an engineering but also medicine background. So I'm really pleased to be able to talk to you all today about some of the work of, it's a really exciting time to be in the field of RNA vaccines, but I think we really need to think about where do we go from here and what is the next generation of this technology. So just to give you an outline, I'll give you a little background for those of you who aren't familiar on self-amplifying RNA. I'll talk to you about the development of the Imperial College COVID-19 vaccine that I was a part of. I'll talk about formulation strategies for RNA and then the future of vaccine manufacturing and my research here at UBC. So I was asked to give some learning objectives for this talk, and so I'll just point these out at the beginning so that you can be mindful of them as we progress through. So I really want to talk about what are the current challenges in the field of RNA vaccines. We have approved vaccines, but they're not perfect. So where are we looking to go from here? I'd like you to understand at the end of this the advantages of self-amplifying RNA compared to messenger RNA, and then some of the strategies that we're doing in my lab, but in the field in general for improving both the safety and efficacy of future RNA vaccines. So I like to start off by just thinking big picture, why do we even need new types of vaccines? We have many licensed vaccines, and for years, these have been working really effectively. So what is the motivation for developing new ones? So we still have a lack of efficacious treatment for many devastating infections, such as HIV or chlamydia. We've recently seen the emergence of multi-drug resistant bacteria. We're, of course, looking to improve the safety profile of our licensed vaccines. So, the mRNA COVID vaccines may soon fall into this category. We're one that's very near and dear to us all now is disease outbreaks. So how can we make vaccines quickly when a new pathogen is discovered? We need to consider the changing age structure of the population. So as people are living longer, their immune system responds very differently. And so we need to kind of understand how we can effectively vaccinate the elderly. And of course, reducing the cost and time required to produce vaccines. So the work that I do really falls within these two categories. So being able to respond to disease outbreaks, but also reducing the cost and time required to produce vaccines. So I think everybody will be familiar with all of this, but I actually like to show this slide because I think it's very emblematic of how much RNA vaccines and nucleic acid vaccines have changed just in the past few years. So I started presenting this slide in 2016 as part of my presentations, and I challenge you to dig and really understand what has already changed, like what's wrong about this slide, what has already changed since the pandemic. So just to give you an overview of the type of vaccines, I think most people are very aware of these, but just to classify these. So they're divided here into replicating and non-replicating vectors. So on the replicating side, we have replicating vectors and live attenuated viruses. On the non-replicating side, we have non-replicating viral vectors, inactivated viruses, virus-like particles, subunit protein vaccines, and at the very end, nucleic acid vaccines. And you'll notice that these are on a spectrum of safety and efficacy where they go in opposite directions. And so what's already wrong about this is that historically it was believed that nucleic acid vaccines would be really high on the spectrum of safety, but really low on the spectrum of efficacy. And what we've really seen throughout the pandemic is that we can have actually really safe and effective nucleic acid vaccines, which I think is why there's so much excitement around this vaccine platform now. Having an efficacy against the original wild type strain of 94 and 95% is pretty unprecedented for a vaccine. And nobody had ever done phase 3 trials for RNA vaccines before that, so it was really exciting for the field overall. So I want to put into perspective why we're interested in using self-amplifying RNA for vaccines. So just to orient you, in the field of nucleic acid vaccines, there's really been 3 types of nucleic acids that have been used so far. So we have plasmid DNA, which is a double-stranded circular construct, usually around 7,000 bases. mRNA, which is structurally very dissimilar to DNA, of course, which is single-stranded and linear, depending on your antigen. It's usually around 2,000 nucleotides. And self-amplifying RNA, which is structurally similar to mRNA, but much larger. So it's usually around 10,000 nucleotides. So compared to plasmid DNA, saRNA doesn't require penetration into the nucleus, so it's a little bit easier to deliver. And there's no risk of integration into the host genome. So this is very advantageous from a safety perspective. Compared to messenger RNA, because it has the self-amplification properties, which I'll talk about on the next slide, this leads to exponentially more copies of the RNA in the cytoplasm, thus you get higher protein expression and you can use a lower dose of RNA. So how is saRNA different from mRNA? So this is a pretty simplified schematic, but really drives home the main difference here, which is on the right for messenger RNA. Once it enters the cell, the mRNA engages with the ribosome and you get translation of your antigen. On the other hand, for self-amplifying RNA, it starts out very similarly. It engages with the ribosome, but you get translation of 2 different proteins. Actually, there are 5 different proteins, but one of them makes this replicase enzyme. So you do get some translation of your antigen, but you get translation of this replicase that then goes back and engages with this original strand of saRNA, such that you get multiple thousands of copies of your original strand of RNA and thus more translation of your antigen. So this usually means that we can use about 100 times less saRNA than we do for mRNA. So to put that into perspective, for the Moderna vaccine, which requires 100 micrograms of mRNA, we could potentially be only using 1 microgram of saRNA. So one of the biggest challenges in the field is the delivery. So how do we get this large negatively charged molecule into a cell? So one of the priority areas in the field that really motivates the use of saRNA is really minimizing the dose of RNA in order to reduce the side effects. So because of the pandemic, we have so much amazing data on dose scaling and the modified nucleotides and lipid nanoparticles in a really large population. And this is data from one of Moderna's initial clinical trials. And there's a lot of data here, but I think it's actually pretty straightforward to understand. So essentially what they did in this study is they were looking at 3 different doses of RNA, 25 micrograms, 100 micrograms, and 250 micrograms. And this was part of the safety aspect of their phase 1 clinical trial. And they characterized the side effects as mild, moderate, and severe. And what I think is really evident from this is that as you increase the dosage, you see an increase of all of these symptoms or side effects that is directly proportional to the dose of RNA. And we've seen this now a number of times. You get this type 1 interferon response into RNA because your body is really adept at sensing foreign RNAs. And so really one of the priorities in the field is minimizing the dose of RNA that's required for mRNA vaccines in non-pandemic contexts. So this means, if we're going to make an influenza vaccine, it's probably going to have a very different safety profile than the COVID vaccines, which were acceptable in a pandemic, but people probably don't want to get a vaccine that can knock you out for 1 to 3 days, potentially annually. So going back to the delivery vehicles, this is one of the most exciting areas of the field. And for those of you who aren't familiar with the different delivery strategies here, I have just kind of an overview slide for this. So for self-amplifying RNA specifically, this is very similar for mRNA. The paradigm in the field is that you have your negatively charged solution of RNA, you complex it to a positively charged delivery vehicle. This is put into nanoparticles, which are most commonly made through a microfluidic system. And the resulting nanoparticle has the RNA encapsulated on the inside. So for saRNA, there's really been 3 different types of delivery vehicles that have been used: polymeric nanoparticles, lipid nanoparticles, which are similar to those used in the approved mRNA vaccines, and nanoemulsions. So this is what I was working on as a postdoc at Imperial when the COVID-19 pandemic hit. And we actually were planning on doing a first-in-human clinical trial for our saRNA that year anyways, although it was a CEPI-funded project, and so it was intended to be against Marburg and rabies viruses. But I want to talk about the development and just why this technology is so advantageous. So just to walk you through this, sure people are very familiar now, but just how you make an RNA vaccine against a brand new pathogen. So this is a schematic of the SARS-CoV-2 virus with the spike protein on the surface of it. We take that spike protein and encode the pre-fusion stabilized spike antigen in our self-amplifying RNA. For our clinical trial, we encapsulated this into lipid nanoparticles in collaboration with Acuitas Therapeutics and then started our preclinical studies. One thing that is really remarkable about making these vaccines is just how quickly you're able to produce and move into clinical trials with RNA vaccines, which is really unprecedented compared to traditional types of vaccines. So I want to walk you through the timeline for our team specifically. So on the 10th of January, 2020, the circulating strain was sequenced and made publicly available. Our team first accessed that on the 20th of January. Very coincidentally, I flew to MSL to interview for the position that I have now on the 21st of January. So it was an extremely busy time, as you can imagine. We ordered our DNA oligos to make our new construct on the 22nd of January. We assembled the genes on the 28th of January. We confirmed expression from our saRNA vector on the 12th of February. We started our immunogenicity studies in mice the next day on the 13th. Because this was a first-in-human clinical trial, we had to start with a toxicology study, which we started on the 30th of March. We submitted our paper on the preclinical work on the 22nd of April, and then started our clinical trial on the 19th of June. So even as a small academic group, we were able to go from designing a brand new RNA vaccine into clinical trials within 6 months. And Moderna reportedly did this in even less time, in just 66 days. So it was about 2 months from start to finish of making and being able to test the vaccine in a human clinical trial. So just to give you a little bit of background on the immune response that we saw with these vaccines. So the first thing that we wanted to do was look at the antibody titers. So this is some of our preclinical work looking at SARS-CoV-2 specific IgG as determined by ELISAs. So these mice were vaccinated at 0 and 4 weeks, and we took serum samples at 2, 4, and 6 weeks to quantify the antibodies. We have a number of groups here, so I'll walk you through all of them. So we use electroporated DNA as our control at the time because there was no good standard mRNA vaccine that had been shown to work as a positive control in the field. And this electroporated DNA was kind of always our baseline for comparison. We used rabies control as a negative control to show that it was actually specific to SARS-CoV-2. We used 4 different doses of our saRNA formulated in lipid nanoparticles, 10, 1, 0.1, and 0.01 micrograms. And then we compared this to the antibody titers in recovered COVID-19 patients. So what you can see here is that we get a pretty good response from the DNA. We were at about 10 to the 5th nanograms per mil for our antibody titers. And with our RNA, it was over an order of magnitude higher. So we're getting just above 10 to the 6 nanograms per mil. There's a really nice dose response curve. And even at our lowest dose of 0.01 micrograms, we're still seeing a higher antibody response than we were seeing when we compared to the convalescent sera from patients. This was I'll caveat this by saying this was very early on in the pandemic. And so these were just the samples that we were able to get from the hospital at the time, although there's been much more work done to compare this to the natural infection now. The next thing we wanted to do was really look at the functionality of these antibodies. So, not just high titers, but are they actually able to neutralize the virus? So I'm presenting here a pseudovirus neutralization assay. So the y-axis is the IC50, but we also did the wild type assay with the wild type virus, and it's really well correlated. So I'm just showing you the pseudo-nukes today. So again, we have our electroporated DNA, and we saw an IC50 of about 10 to the 3rd. Our RNA, again, was even higher, which kind of directly reflects the antibody titers that we saw. So at about 10 to the 5th for the IC50 for our highest dose of 10 micrograms. And even at our lowest dose, we're still seeing a really good neutralization, again, higher than that in recovered COVID-19 patients. We wanted to look at the correlation between the amount of antibodies and the extent of the neutralization. So we did this for both the mice and the recovered COVID-19 patients. And you can see these are both positively correlated. So what this tells us is that not only are you making more antibodies, but they're functional as well, which is indicative of a good immune response. We also wanted to characterize the cellular response. So we did this using an interferon gamma ELISpot assay, where we take out the splenocytes after 2 weeks after the final vaccination. We re-stimulate them with peptides from the spike antigen and then quantify how many of those splenocytes are expressing or secreting interferon gamma. So this is quantified on the y-axis as the spot forming units per million splenocytes. Again, we have our electroporated DNA, and it may look like there's not really a response here, but actually 250 spot forming units per million splenocytes is actually a pretty normal and pretty good response for a DNA vaccine. It's just really eclipsed by the cellular response that we saw for the RNA. So for our highest dose, it was up to 250 spot forming units per million splenocytes. And even at our lowest dose of 0.01 micrograms, we still saw over 1,000. So not only are we getting a really good antibody response, but we're getting a really excellent cellular response as well. So people always ask me, where is the Imperial College siRNA vaccine now? So we finished our combined phase 1-2 clinical trial in January of 2021, right as I was starting at UBC. And at that time, there was already the AstraZeneca and some mRNA vaccines that had been recently approved in the UK. And so the government didn't want to fund the phase 3 trial for another 130 million pounds, which is very understandable. So the team is now targeting different mutations and looking at booster doses. We've done some work with AstraZeneca, actually with the ChAdOx vaccine, to look at heterologous vaccine regimens. We started a company based off this platform, which is called VaxEquity, and really excited to announce last year that we partnered with AstraZeneca to really develop this platform. So coming back to the delivery vehicles and where we go from now, there's a good proof of concept here and it's been in the clinic, but how do we make this even better and really understand more mechanistically how these RNA vaccines are working and why? So just to jog your memory, there have really been 3 different types of delivery systems for self-amplifying RNA. And today I'm going to talk about 2 specifically that we did a more mechanistic and head-to-head comparison for. So I want to start off by thinking really about what is the importance of the delivery vehicle. So it's not just getting the RNA into the cell, but I always think back to this schematic that was published by Norbert Hardy in 2018, who worked really closely with Drew Weissman. And you can see in here, this is all of the pathways that we know by which RNA is sensed by the cell, right? So we have all of the TLRs and the endosomal sensing mechanisms, as well as the cytosolic RNA sensing mechanisms, common ones like MDA5 and RIG-I, and this is well understood and how this plays into the interferon response. But what always sticks out to me from this figure, and I think we still don't have great answers for, is over here to the right, where it just says carrier sensing ? So we know that these delivery vehicles are being sensed by cells, but we don't really know how or what effect this has on the vaccine immunogenicity. So the point of this study was to really compare 2 of the leading delivery vehicles in the field. And this actually came from a paper that was published by Dan Anderson's lab in Nature Biotechnology just a few weeks ago. And it really spells out the state-of-the-art delivery vehicles. So for the lipid nanoparticles, these consist of 4 different lipids. So an ionizable lipid, a helper lipid, cholesterol, and a PEG lipid. And they really nicely illustrated on the left here. So MC3, as it's called, is actually the approved siRNA drug called Onpattro. SM102 is Moderna's lipid, and ALCO315 is Acuitas's lipid that was used in the Pfizer and BioNTech vaccine. There's 2 different helper lipids that people typically use, DSPC and DOPE, and then PEG-DMG is the most common lipid, although Acuitas now has this proprietary PEG lipid that they use. And so these are really, I think, kind of the leading delivery vehicles for mRNA in the field, which is LNPs and then polymeric systems. So our goal here was really to do a head-to-head comparison of polymeric and LNP formulations to understand more about the how they work and why that is. So this was work that we did in collaboration with Molly Stevens' group at Imperial. So we developed a polymer specifically for siRNA delivery, which is called PABLE. And PABLE is a bioreducible cationic and linear polymer. And you'll notice that these particles are very simple. They just have 2 components, which is the PABLE and the self-amplifying RNA. And on the right, we have the lipid nanoparticles, which, as I said before, contain 4 different lipids: an ionizable lipid, phospholipid, cholesterol, and a PEG lipid. And for this study, we used 2 different phospholipids, DOPE or DSPC, and these will be annotated throughout as PE or PC next to the formulation. And this work was actually done in collaboration with Precision Nanosystems here in Vancouver. So the first thing we wanted to do was really characterize the formulation. So we looked at the size, the charge, and the encapsulation efficiency for all of these formulations. I'll walk you through this here. So on the right, we have PABLE, which is our polymer, and then 5 different lipid nanoparticles. So LM just stands for lipid mix, and we have 1 through 4, and then this is formulated either with DOPE or DSPC. What you can see from the size is that they're all very similar in size, about 80 nanometers with a really good polydispersity index of 0.1. So this is within the target product profile that we typically be looking for. For the zeta potential, we see that the PABLE particles are positively charged, whereas the lipid nanoparticles are neutrally charged, which is really the main difference between these formulations, and we're starting to dig more into now. For the encapsulation efficiency, they all have really good encapsulation efficiency. PABLE particles don't require any downstream purification, so they have 100% encapsulation efficiency, and all of the LNPs were above 84%. So the first thing we wanted to do was look at the in vivo protein expression from all of these formulations. So again, we have our PABLE and the 5 LNPs. We use luciferase as a reporter protein in this case, and we do an intramuscular injection and then quantify how much total flux we see from each muscle. And what you can see here, we have these statistics, but I think this is a graph that really speaks for itself, where we see much higher protein expression intramuscularly from the PABLE particles compared to the lipid nanoparticles. So our PABLE is just over 10 to the 6th, whereas our highest lipid nanoparticle formulation is just over 10 to the 4th. So we're seeing about 100 times as much protein expression from the polymer PABLE. And within the lipid nanoparticles, you can see that all the formulations that had DOPE worked much better than the DSPC in this case. So we wanted to go on and then see how all of these formulations compared in a vaccine model. So we used HA, hemagglutinin of influenza, as a model antigen. And the first thing we did was characterize the antibody titers that we saw against HA, IgG, using an ELISA. So again, these mice were vaccinated at 0 and 4 weeks, and we quantified the antibody titers at 4 and 6 weeks. You can see with our PABLE that we see a pretty good immune response. It's almost 10 to the 5th nanograms per mil, but the LNPs really blew the PABLE out of the water, right? So with our highest performing LNP, it's just over 10 to the 7th nanograms per mil. So even though we're seeing higher protein expression with the PABLE, the LNPs are a much more effective vaccine, which is really quite perplexing, because you would assume that if there's more protein expression there, that would confer greater immunogenicity. We went on to challenge these mice, and in the preclinical model, the body weight is reflective of the response and protection against the virus. So we have our naive group, which you can see that by day 5, all of those mice had met the endpoint for body weight. Whereas for the PABLE group, they all survived, but you can see that at the peak of the infection, they got down to about 90% of their original body weight. So they weren't completely protected, which is really directly reflective of the antibody titers. For the lipid nanoparticles, the ones that had the highest antibody titers, we saw no change in the weight in these mice. So in this case, the antibody levels really conferred the protection against the influenza challenge. And again, the formulations with DOPE had consistently higher levels of protection than the DSPC. We wanted to look then how the route of administration affected the immune response. So in this case, we used the SARS-CoV-2 as a model antigen, and we looked at both the systemic and the mucosal IgG. So using just our highest performing lipid nanoparticle here, which was lipid mix 3 with DOPE, we started with a dose titration. So our highest dose was 1 microgram, and then going down to as low as 0.0001 microgram or 0.1 nanogram. So really small dose in this case. We compared this to 1 microgram of PABLE, our naive group, and then 1 microgram of the LNPs, but instead of injecting intramuscularly, we administered it intranasally. And this answer for us, does the immunogenicity vary by antigen? Even with SARS-CoV-2, we still see a really good immune response. So really nice dose response curve here with a systemic IgG just over 10 to the 5th nanograms per mil with our highest dose of 1 microgram. And even at our lowest dose of 0.1 nanogram, we're seeing some antibody response, although there was a lot more variability at this lower dose. Even at this lower dose, this is pretty comparable to the 1 microgram of Pable. So these LNPs are really potent. When we administer them intranasally, we see a good antibody response, but it's about 100 times or 10-fold lower than what we see with the intramuscular injection. And I will caveat this by saying that these weren't optimized for intranasal administration. We just wanted to see if we were still getting good systemic and mucosal antibodies when we gave it this way, but it's about 10 times lower. So with the mucosal IgG, we do see some antibodies here at the mucosal surface, although you'll notice it's a lot lower than what we see systemically. So we saw it for all of our groups, although again, a lot more variability at our lowest groups here. And with the intranasal administration, it didn't really change kind of that ratio. So it was still much higher mucosal antibody levels at with the intramuscular injection. So we then went on to characterize the neutralization. And again, this really reflect the antibody titers. This is pretty obvious now. So as far as the IC50 goes, with our highest dose, we see just over 10 to the 3rd for the IC50, down with our lowest dose where there was so much more variability with the antibodies, we weren't really seeing any neutralization. And even when we gave 1 microgram of LNP intranasally, we still see a pretty good neutralization, although again, it's about 100-fold lower than the LNPs when they're administered intramuscularly. So again, we wanted to characterize the cellular responses with these. And so we did an interferon gamma ELE spot. So on the y-axis is the spot forming unit per million splenocytes. With our highest doses of LNPs, we see just over 1,000 spot forming units per million splenocytes. And even at our lowest doses, we're still seeing a pretty good cellular response here. So at the lowest dose, we're seeing about 100 spot forming units per million splenocytes. We see an okay cellular response for our Pable particles, and they're quite similar actually in this case to the lipid nanoparticles that were administered intranasally. Although you notice that here, both the cellular and humoral immune responses were quite muted compared to the intramuscular injection. So this is really where we get into the mechanistic work for understanding how these formulations are being sensed and expressed by cells. And we're still digging into this today, but when we looked at this, we wanted to get a sense of the reactogenicity, or like how cells are detecting the lipid nanoparticles and Pable. So to do this, we measured the systemic cytokine levels 4 hours after administration, and we did a Luminex panel of all these different cytokines. So they're not all presented here, but just to tell you, we didn't see any detectable levels for GM-CSF, interleukin-1 beta, IL-2, 3, or 2, 13, or 18. And you can see some differences here with the groups we compared. So with all of these different cytokines, we compared 1 microgram of LNPs administered intramuscularly with either LNPs or Pable. We also compared LNPs administered intranasally and a naive group. So you can see for the interferon gamma, there's a slight increase compared to the naive group, although it wasn't statistically significant. There's also a trend of increasing IL-12. All of the IL-4 was increased compared to the naive for all of the treatment groups. But really the main difference here is the IL-6. So that you see for the lipid nanoparticles that were administered intramuscularly, we see a profound increase in the IL-6 compared to the Pable or LNPs administered intranasally, which weren't different from the naive group. So there's really something in this IL-6 activation that seems to be really important for the LNPs and their vaccine immunogenicity. So I want to think now about where are we going, and just to kind of highlight how powerful this technology, just thinking about how we make vaccines, as I am a chemical engineer by training, right? So just to highlight, manufacturing of vaccines hasn't changed a lot until very recently. So this is a picture of making the annual flu vaccine in 1957, and then how we still make it in 2021. So this is produced in eggs. It's a very manual labor process and takes a long time to complete. Other protein and viral vaccines will be produced in these large bioreactors where you have to grow cells, wait for them to produce enough protein or virus, and then purify that out, which again is a really time and resource intensive process. On the other hand, for RNA vaccines, we can make about 1 million doses in just 100 milliliters. And so this is a very modular technology, and there's many instruments and kind of scale up now for the formulation like that from Precision Nanosystems, where you can easily make these lipid nanoparticles on a lab scale and then scale them up for the clinic as well. So to put this into perspective, I like to think about how many Olympic-sized swimming pools it would take to vaccinate the entire world with an RNA vaccine. So I did this calculation specifically for the Pfizer and BioNTech vaccine. So if we assume we have 7.7 billion people total, and each initially needed a dose of only 2 doses, this comes to a total of 15.4 billion doses. Each dose is 0.3 milliliters. Thus, the total required volume is 4.62 million liters. Each Olympic-sized swimming pool has a volume of 2.5 million liters, and so this means that you just need 2 Olympic-sized swimming pools to vaccinate the entire world. And while this is an incredibly large amount of liquid, it is actually feasible, right? So this is why we've seen now that billions of people have been able to get an RNA vaccine, and this manufacturing is really unprecedented compared to, protein or viral vectors, which are much more limited. So I have a vision of how the manufacture of vaccines is going to change over time. And just to tell you our story of how little infrastructure there was and still kind of currently is for manufacturing RNA vaccines. For our clinical trial, we got our RNA from California. The lipids came from Acuitas here in Vancouver. These were shipped to Austria to be formulated. And then the formulation was shipped to Italy to be put into vials and then shipped back to the UK to start our clinical trial. And what's really amazing is none of this was actually made in the UK for our clinical trial. But because RNA is so modular and really doesn't require this huge manufacturing footprint anymore, I think in the future we're going to start to see more regional manufacturing centers where you can make vaccines in an outbreak if you need, but also for a seasonal influenza vaccine. But the same technology could be used to manufacture personalized cancer vaccines as well. And so I think we're going to start seeing this pop up more locally, which I think also just inherently makes it more equitable. So in conclusion, today I've shown you that with our SARS-CoV-2 vaccine, we see that our vaccine induces higher antibody titers and neutralization than a natural infection in recovered COVID-19 patients. We completed our phase 1 and 2 clinical trial for an saRNA vaccine, and our team is now targeting mutations and boosters, and I'm a co-founder of VaxEquity. For the formulation strategies, the main takeaway is that the polypeptides had 100 times higher protein expression, but the lipid nanoparticles had 100 times higher immunogenicity. So we're working to understand why that is and if we can make even better delivery systems. And I think for the future challenges, we'll really see this modular manufacturing of RNA vaccines that will enable a global response to future outbreaks. So to give you an idea of what we're doing in the lab, I think it's a really exciting time to be in the field. The plane is really off the ground, but I think you really need to be thinking about what is the next generation and how do we make these vaccines even better. So our lab does RNA molecular engineering. So how do we engineer the RNA to be more stable and have higher levels of translation? We're also really interested in biomaterials design. So how can we use these particles to get more of the RNA to the cells and tissues where it actually needs to be? And we're also thinking about how do we make better vaccines and use RNA for other applications. So we're interested in making other vaccines like flu and chlamydia. We're seeking to gain a better understanding of the immune response, but we're also looking at using RNA to express monoclonal antibodies, which could be used for treating cancer or preventing disease. So I was asked as well to come up with some questions and opportunities for collaboration just to get people thinking. So some of the things that I thought there would be really nice overlap with is I'm really passionate about making clinically translational technologies. So One of the main challenges in the field is that we do most of this optimization in mice, and we're really interested in testing this in human tissues, as I think this is one of the main challenges in the field. Is we optimize it in mice, their immune system is very different from humans. And so we are actually working with some plastic surgeons now to get skin tissue, but we'd be interested in testing different types of human tissues as well if we're able to have access to this. And I think this is actually really integral in the development of new RNA vaccines and especially therapies. We're always looking for new vaccine or therapy targets. So if you have a disease where you've been treating it with a protein and you want to do it with RNA instead, you can very easily clone and make new constructs. And also, eventually looking for collaborators for clinical trials of promising preclinical candidates. So with that, I'd like to thank everybody that was involved in this work and my lab at UBC. So our collaborators at Imperial College London, Precision Nanosystems, and Acuitas, as well as our funding sources here at UBC. And with that, I'll take any questions. And my email address is at the bottom. So if you have any questions or want to reach out at any point in time, please feel free.